Vaccination or immunization, both mean the same thing. Vaccination provides artificial adaptive immunity to a pathogen without the dangerous exposure to the pathogen due to an infection. Vaccination is exposure to a safe form of the pathogen or a piece of a pathogen or a very low dose of a pathogen. A safe exposure can induce an adaptive immune response and immunological memory. A later exposure to the infectious pathogen leads to a secondary immune response due to the presence of the memory B and T cells. The pathogen is eliminated before illness sets in because secondary responses are rapid, specific, and strong. You can click here for an article about vaccine technology. The chart shows the concentration of the produced antibodies, over here on the y-axis, in response to a pathogen as a function of time down here. So let's say over here we have an initial exposure to the vaccination, which is trying to protect us from the pathogen. It takes quite some time to build up the response, and then it peaks right here, not terribly high, and this is the primary immune response. Then, if you have a secondary exposure to the pathogen, let's say this is the real pathogen hitting you and it's not the vaccine, look how quickly it ramps up, much quicker than this time over here. Here it's a little bit, just a little bit there, a lot smaller, and the amount of antibodies produced is much, much higher. That's the secondary immune response. So let's go back to this paragraph here. Without vaccination, a pathogen would induce a primary response 7 to 14 days after the infection. This delay could give the pathogen an opportunity to evade the immune system, reproduce, and establish residence in the body. The secondary response is much quicker and produces more antibodies. A vaccination is superior to passive immunity because vaccination leads to the development of a lasting adaptive immunity with the involvement of APCs, T and B cells. The vaccination causes the APCs, over here, to process injected antigens and present them to the T cells. So here's our injected antigens. They're being presented to the T cells, which then start the adaptive immune response. The next step, when the APC presents right over here, okay, there's the antigen to the T helper cell, it then communicates with B cells and cytotoxic T cells, causing them to respond to the artificial antigen. Finally, memory B and T cells develop. While they recognize the injected non-infectious antigens in the vaccine, they also provide long-lasting immunity to the natural pathogen. Later exposures to the natural pathogen will result in rapid, strong secondary immune responses. Here's the memory cells persisting in the body, and here's the antigen-specific antibodies made by the memory B cells to that pathogen. And over here, these are our pathogens. When a large percentage of a population has been vaccinated for an infectious disease, that population has herd immunity, talked about that earlier, a status that prevents the spread of the disease. Because most people have immunity to a pathogen, they will be less likely to pass it on. When herd immunity is achieved, those who cannot be vaccinated due to special health status or choose not to be vaccinated may still be protected from the disease. Long before modern vaccine development, many societies in Africa, China, and Turkey knew that if they exposed the person to a tiny dose of cowpox, which is called by the vaccinia virus, that person would have immunity to dangerous smallpox, right? Cowpox, then smallpox, which is caused by the closely related variola virus. The viruses that cause the two diseases share many antigens. While cowpox is not dangerous to humans, the virus that causes it shares many antigens with variola. Scratching the skin with infected cow cells is enough of immunization to protect most humans from smallpox. The scientific concept underlying modern vaccines is the same as that which was applied to smallpox. However, most vaccines are much more difficult to develop. Medical research has led to vaccines that can protect us from over 40 different infectious diseases. For more information, please visit these two links. Eradication of any disease from a region of the world, however, requires herd immunity, which may be difficult to achieve due to social factors. So while many diseases have been prevented in many countries, most of these diseases persist in at least some parts of the world. For example, while polio has declined by 99.9% over the past 35 years due to worldwide vaccination, it still occurs in Afghanistan and Pakistan, where the vaccine has not been available to everyone. So here's our types of immunity that we've gone over. Vertebrates are born with innate immunity, over here, and it is nonspecific. Adaptive immunity is antigen-specific and be obtained through natural means, through the mother or being infected, and through artificial means, which are antibody transfer and immunization. So here's our artificial, and here is the natural. Some vaccines are very difficult to develop due to challenging characteristics of the pathogen. Here's our first example. In spite of 40 years of efforts by laboratories all over the world, no vaccine for HIV has been developed. The challenges presented by HIV vaccination development are HIV has a very high mutation rate, so it changes its antigens faster than the immune system can keep up with it. It also establishes asymptomatic infections, so that people can pass it on to others for a long time before they know they even have it. And HIV establishes infection in the DNA of host cells, a location that is protected from the immune system. Example 2. Malaria vaccines are difficult to develop because the parasite Plasmodium has a very complex life cycle and has another animal host, the mosquito. It hides inside red blood cells, which are inaccessible to the immune system. The natural immune response to malaria is poorly understood, which makes it hard to control with the vaccine. Because each pathogen has unique characteristics and each immune response is also unique, each vaccine must be a unique product. Some vaccines induce immunity to a dangerous chemical produced by a type of bacteria, like the tetanus vaccine. Some vaccines induce immunity to a virus particle, like the polio vaccine and the smallpox vaccine. Others are subunit vaccines, which give immunity to a piece of a pathogen, such as in the influenza vaccine or the flu and COVID-19 vaccine. The vaccine for COVID-19, which is caused by a coronavirus called SARS-CoV-2, is a subunit vaccine. It provides immunity to one antigen on the virus particle, the spike protein. It is safe because the vaccine has no virus in it, only the genetic code for a version of one protein. It was developed very quickly because the virus does not mutate as rapidly as HIV or influenza. Scientists were able to identify an antigen that induces strong immune responses, the spike protein. A few biotechnology companies had the needed technology in place before the pandemic arose. Natural immunity to COVID-19 that results from an infection differs from vaccine-induced immunity to COVID-19. Natural immunity produces immune responses to more than one viral antigen. The vaccine only produces immune response to the spike protein. Some people who have been immunized for flu and COVID-19 still contract these viruses and become ill. Some viruses mutate very quickly, and if you are immune to one strain of the virus, you may still experience symptoms if you become infected with a different strain of the virus. But being immunized does improve outcomes of infections. That's very critical. Immunized people are less likely to become seriously ill or die, or to pass the virus on to others. It is still good for the individual and the community to get immunized to viruses that change over time. Vaccines use many strategies to induce a protective immune response and lasting immunity. And here's a quote from this paper here by Lee et al., so feel free to click on that for the full article. Mathematical models project that vaccines will prevent 23.3 million deaths from 2011 to 2020, compared to what would have occurred if there were no vaccines available. That's incredibly impressive. Vaccines are tested extensively before they are given to the community to be sure that they are safe and effective. They work best for everyone if herd immunity is achieved. Only 2 infections, smallpox and rinderpest, have achieved herd immunity for the entire human population. New diseases will continue to emerge, and vaccine development will also continue.